Oh, hi. I didn't see you there. Well, since you're here, me and Bunny are gonna explain to you about plant immunity. Right, Bunny? No? Fine.
The initial defense of the cell is the cell wall, which consists of cellulose, and a waxy cuticle, which is a film that covers the surface of leaves. In the roots, the Casparian strip acts as a waterproof band in the endodermal cells to prevent penetration from pathogens.
Okay.
So here we have our PRR, our pathogen recognition receptor, and here we have our PAMP, our pathogen-associated molecular pattern that are expressed on pathogens. And when a pathogen is invading the immune system of a plant, the PRR recognizes the PAMP, or sometimes they don't recognize it and get attacked, but sometimes they recognize it and attack it, like so, and then they can kill off the pathogen. So what happens when you have a stronger pathogen? Well, sometimes pathogens can insert an effector protein into the host cell, and the effector can actually inhibit the PTI response. But plants have also adapted another way to overcome this, and that's by the LRR, the leucine-rich receptor. And what happens in the ETI response, the effector-triggered immunity, is that the LRR can recognize and bind to the effector protein, and when recognized, they can signal to the infected cell to die.
BAM! Apoptosis. I feel like I just need to put that
Oh no.
Did you restart?
Yeah, could go.
When the cell wall and waxy cuticle fail to keep plant pathogen out of the cell, the invasive pathogen will enter the intercellular space and seek to further the infection inside of the cell membrane. In order to defend itself, the molecules released from the pathogen onto the membrane, such as lipopolysaccharides, flagellin, and chitin, are the pathogen-associated molecular patterns that are bound to pattern recognition receptors, or PRR. The PRR interacts with a central regulator known as racinosteroid insensitive 1 associated kinase 1, or BAC1. The association with BIC1 begins a signal cascade through MAPK toward the nucleus. The transcription of new proteins creates complexes which limit the colonization of the pathogen and sends a distress signal before the plant cell ultimately dies to other cells. This is the PTI response. With a more potent pathogen, a bacteria cell via pilus or fungus via Haustorium is able to penetrate the plasma membrane and place effector proteins in directly. These effector proteins are able to inhibit the PTI response. The plant then binds nucleotide binding leucine-rich repeat receptors to the effector proteins, prompting another signal cascade toward a distress signal in the nucleus, and ultimately cell apoptosis or cell death. This is the ETI response.
The PTI receptor protein for pathogens, or PRR, lies on the extracellular space of the cell and is flagellin sensing. The active complex that is formed between BAC1 and PRR after a PAMP is bound to the PRR causes that phosphorylation via BIC1, MAPK, or CDBK leads to the production of phytohormones, such as salicylic acid, jasmonids, and nitric oxide, for defense mechanisms such as stomata closure, distress signal, N2 fixation, and nitric oxide. But unlike the PTI receptor protein, the ETI receptor protein, which consists of a toll interleukin receptor, a nitrogen binding protein, and a leucine rich portion, the ETI receptors within the cell have 3 ways of binding to a pathogen effector protein. The direct method involves the effector protein binding to the leucine rich portion directly. The guard/decoy portion involves the binding of an accessory protein that has been infected by an effector protein to the leucine rich portion. And finally, the bait method involves the binding of the accessory protein being infected, the effector protein itself, to the leucine rich portion, which ultimately leads to cell death.
Well, we sure learned a lot about plant immunity, didn't we, Bun Bun? Yeah. No? Okay. Stay classy, plant physiology.